Okay, so for this week, we will be doing the lesson on hereditary. And the first thing we're going to do is to label these diagrams for the different stages of meiosis. Okay, so meiosis has two divisions. There's meiosis 1, and then there's meiosis 2. So over here, the first picture, it is telophase 2. And you can see it's telophase because now you pretty much have 4 cells. So it's an indication that it is the last step of the meiotic division. Okay, so over here, this is actually 2 separate cells. And in each of the cells, the chromosomes are being pulled to the opposite end of the cell. So we know this will be anaphase 2. This is one single cell, and the chromosomes are being pulled to the opposite end. And so this is anaphase 1. Chromosomes are still in the middle. We see only one single cell. So that would be prophase 1. Over here, we have a cell that is dividing. And you can see the chromosomes are kind of going back into the nucleus. That would be the telophase number 1. You can see all the chromosomes lining up in the middle. So that is metaphase, metaphase 2. We have all the chromosomes lining up in the middle. That is metaphase 1. Right? The difference between metaphase 1 and 2 is that in metaphase 2, you can see it's 2 separate cells doing the same thing, and here is just one single cell. And finally, this is prophase 2. Okay. So for those of you who just joined, I'm gonna let you catch up a little bit. Okay, maybe take a picture, do a screen grab, and we'll continue. Okay. All right, next question has to do with the number of chromosomes. So let me just remind you what meiosis looks like. So we start with a single cell, and we're gonna have the maternal chromosome. Okay, I'm just gonna draw 2, and then we're gonna have the paternal chromosome. Okay, so this is chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from mom, chromosome 2 from dad. And so these chromosomes, I should probably do it a little bit better. These chromosomes, they are called homologous chromosomes. Okay, so they are the same size, same length. And then they have genetic information in the same order. Okay. So this would be a germ cell. A germ cell is a cell that is found in the gonad, ovary or testicles, where it will undergo meiosis to produce sperm or egg. So what will happen is, okay, we will start with like 4 chromosomes here. Okay, 4 chromosomes. 1, 2, 3, 4. 4 chromosomes. Okay, and then we have DNA replication. Okay. So after DNA replication, what's going to happen is we will have 2 of them stuck together. like that. Okay, so this would be kind of like prophase number 1. Okay. So how many chromosomes are here? Well, this is the confusing part, right? And I got confused last week too. Yeah, Paul is correct. It's 4 chromosomes. Okay, as long as they are stuck together, it's considered as 1. Okay, so it's still 4 chromosomes. Okay, but we would have like 8 chromatids, right? TID, right? Okay, so let's label some of these things. Okay, so these 2 things that are stuck together at the centromere, right? Those are sister chromatids. Okay. And what do we call this whole thing here? What do we call this whole set? The 4 of them all together. Anybody remember? That is okay. Yeah, we have 2. We have the red and the blue are the homologous chromosomes. Yeah. But when they're all together like that, right? This is called a tetrad. Okay. Tetrad. 4 sister chromatids together. Okay. So one of the things that happens in prophase is that there is going to be this exchange of genetic information, right? Crossing over, right? We call it. So, you know, what's going to happen basically is this. Let me try to draw this for you. I'm sure you've seen it in the lecture notes. Okay. Sorry. So a little bit of the blue will go to the red, and a little bit of the red will go to the blue. Okay, like this. Okay. Same thing for the small one. Okay, so this is called genetic recombination. Okay, so crossing over between homologous chromosomes. Generate genetic variation. Okay, that's why we don't, like, our offsprings don't look identical to us. And so this happens in prophase number 1. Okay, so anyway, long story short, these are all pretty much explained in the lecture already, but you know, you're going to have the metaphase and you're going to have an anaphase and a telophase, and what you're going to end up having is you're going to have 2 cells. Okay, 2 cells, and one cell is going to get, you know, one of the big X like that, and then, you know, the other one's going to maybe get a small X. Okay. It's really hard to draw with all these, you know, color stuff going on. Okay, and then you're gonna have a small X here. You're gonna have the big one. Okay, something like that. Okay, the idea is that you will get the number 1 and number 2. Okay, so how many chromosomes here? 2 chromosomes. 2 chromosomes. Okay. And then meiosis 2 is going to happen. And then you're going to split again. You're going to split again. Okay. So these 2 sister chromatids will get separated. Okay. So you're going to get, one cell is going to get one of them, and then the other one's going to get one of them, like that. And then this end will be here. And then same thing for this chromosome. Okay. There we go. Okay. So in the end, we will still have 2 chromosomes. Okay, for both of them. So basically, we started with 4. After first division, we have 2. After second division, we also have 2. So basically, in this chart, right, we started with 46, which is 23 pairs of chromosome. Okay, so the 46 is diploid. Diploid, 2 sets. Right? 1 set of red, 1 set of blue, so to speak, 1 from each parent. And then after meiosis 1, it is going to be 23 chromosomes, haploid now. Okay, and then 23 again, haploid. How many number of homologous chromosomes do we have at the end? Well, let's take a look here. None, right? There's no homologous chromosome for this one anymore. Right? The homologous chromosome is in the other gametes, right? Okay. So basically, this chart is very simple. It's just the number here, starting number, and everything is going to be half. So this is 18. Everything's going to be 18. And then the first one will be 36. But it's 0 across for the number of homologous chromosomes in the end. This is 45. Then we started with 90. 45, 45, 99, 18. Okay, so everything in the original is diploid. Everything else afterwards, haploid. In order for meiosis to take place, though, the starting number of chromosomes must be even number. Okay, because only even number can be divided by 2 equally, right? If you have an organism with like 7 chromosomes, you cannot have one gamete of 7. You cannot have one gamete with 3 chromosomes and then the other with 4, right? Like it doesn't make sense, right? Okay. So that's that. Any questions before we move on? Pretty straightforward stuff. Okay, let's compare mitosis and meiosis. So for mitosis, the number of divisions is 1. For meiosis, 2 divisions. Meiosis 1 followed by meiosis 2. Number of daughter cells produced for mitosis, it is 2 cells. For meiosis, it would be, well, for male will be 4 sperms from 1 germ cell. For female, it is going to be 3 polar bodies, which will break down, and 1 egg. Okay, so like you have your germ cell, right, 46, and then after the first division, instead of having like 23 like this, you're gonna get 1 very tiny one and then 1 big one. This one's gonna maybe have another division, okay? But these are gonna break down. These are polar bodies. They will break down. Too small, okay? And then the egg is the one that will get most of the cytoplasm. Okay, so this is a mature egg, an ovum, and then these ones here, these things 1, 2, 3 here, those are the polar bodies that will break down. Okay, daughter cells. For mitosis, the daughter cells will be diploid, 2 sets. But haploid for meiosis. Number of chromosomes in daughter cells. If we're talking about humans, and we are, I think, yeah. So we started with 46 chromosomes, and the daughters will be 46 chromosomes. But in the gametes, in the sperm and egg, it will only be 23, which makes sense. 23 from the sperm will merge with the 23 in the egg to create 46 in the baby. Okay, so comparing prophase in mitosis and prophase 1 in meiosis, what is the difference? Okay, so in mitosis, homologous chromosomes do not pair up. Okay, whereas in meiosis, homologous chromosomes form tetrads. Okay, and then crossing over takes place. Crossing over occurs to create genetic variation. Okay. The Christian has a question. What is diploid and haploid? Okay. So give me 1 second, huh? Just gonna Google search an image to help you understand. One second. Okay. So here is a karyotype, right? Remember karyotype? It's like basically all the chromosomes lined up. Okay. So the chromosomes, chromosome 1 here. Sorry. Chromosome 1 here, right? That's from one parent. Let's say it's from mom. Okay, so chromosome 1 from mom, chromosome 2 from mom, chromosome 3 from mom, chromosome 4 from mom, so on and so forth. Okay. And then you also have the exact same thing from dad. Chromosome 1 from dad, chromosome 2 from dad, 3 from dad, 4 from dad, so on and so forth. Okay, so for each chromosome, you have 2 copies. Okay, so in the entire chromosome set, there are essentially 2 sets of chromosomes. Okay, so when you have 2 sets, that's called diploid. Okay. During meiosis, each of the sperm or egg will only get 1 set. Okay, so one of these chromosomes will go to the sperm, and then the other one will go to another sperm. Okay, so together, it's just going to be 23 chromosomes, and there is only 1 set. Right, so this is haploid. Okay, so sometimes you will see people write diploid as like 2N, and then haploid is just 1N. So when the sperm and the egg comes together, right, haploid and haploid, to form the zygote, zygote is the baby. Right, so 23 plus 23, you get back your 46. So now you're 2N again. Right, so this is zygote, which is diploid. Does that make sense? Okay, I hope that makes sense. If you have more questions, please let me know. Okay, metaphase versus metaphase 1. So what's happening in metaphase? So in metaphase, if you think about it, the chromosomes, they line up like this, right? They line up in the middle. Okay, so homologous pairs, or we'll say like tetrads, do not. Sorry. Maybe we'll just say like sister chromatids line up at the center. Okay. Whereas for metaphase 1, right, it is the tetrad that lines up in the middle. Okay, the tetrad lines up. Genetic relationship of daughter cells to parent cells for mitosis is identical. Okay. For meiosis is variant, it's different. Types of cells involve somatic cells, which are like bodily cells, okay, your skin cells, liver cells, whatnot. But for the meiosis, only germ cells. Okay, so these are cells that will become sperm and egg. The function of mitosis could be repairing, like wound healing, for example, growth and development. If you need more cells, you're gonna have to do mitosis. Whereas meiosis, the function is sexual reproduction. Okay, that's the difference between mitosis and meiosis. Any questions? All right, let's do the Mendelian genetics. State the law of independent assortment. Okay, so this is one of two Mendel's law. So the law of independent assortment states that the alleles of two or more genes. Okay. For those of you who don't know what an allele is, an allele is basically different version of a gene. Okay, so if the gene is eye color, an allele would be like brown eye or black eyes. So the allele of two or more genes get sorted into gametes independently. Of one another. Okay. So what does that actually mean? Well, let's say I have one chromosome. Okay, this chromosome from dad, for example. There are 3 genes on it. Let's say, okay, big A, big B, and small c. And then on the maternal chromosome, there is small a, small b, and big C. So let's say we look at a sperm. Okay. What kind of combinations do we have in the sperm? Okay. So they might get big A, small b, and small c. Okay. Just because big A and big B are together on the same chromosome in the original cell does not necessarily mean they have to be together in the sperm. Okay. So whether it gets A, big A, or small a is independent of whether you will get big B or small b. Similarly, whether you get big B or small b is independent of whether you get small c or big C. Okay, each of the allele will get sorted independent of each other. Okay, think of it, it's like flipping a coin. If I flip one coin and I get a head, okay, and then I flip the other coin, okay, whether you get heads or tail on the first coin will have absolutely no effect on whether you will get head or tail on the other coin, right? Okay, what's the probability of getting a head on a coin? You either get heads or tail, right? So getting a head would be 50%. Okay. What about this coin? Right? Another coin, also 50%. So whether you get heads or tail on the first one does not affect the probability of the second one. They are independent events. Right? So same situation for each gene. Whether you get big A or small a, 50-50 chance, right? Does not affect whether you get big B or small b. That's essentially what the law of independent assortment means. Are we okay on this concept? Okay, so here is a question I could ask you. related to independent assortment. How many unique gametes could be produced? Gametes are like sperm and egg, right? Through independent assortment. If the genotype is like big A, small a, big B, small b, big C, big D, small d, and big E, big E. So I'll do a quick poll just to see where you guys are at, and then I'll explain. Okay. Thank you for participating. Let's see. Okay, so let's take a look at each individual pair. Okay, so we have big A, small a. So that means if you think of the gametes, they could either get big A or small a, right? Basically, two choices. Okay, two choices. So I'm going to put 2 here for A. For B, same thing, big B, small b. So you can get a gamete with big B or small b. So, and other 2 choices. Okay, C, well, because it's the same, it's homozygous, big C, then doesn't really have any effects because you're guaranteed to get a big C, right? You're not going to get a unique gametes, right? So I don't have to worry about C. And then for D, right? Big D, small d, 2 possibilities, right? And then I don't have to worry about E either. So the correct answer is 2 times 2 times 2, which is 8. Okay, I'm going to list all the possible outcomes for you. So you can have big A, big B, big D in the gamete. You can have this. And then what else did I miss? This one. Oh, 1, 2, 3, 4, 5, 6. And then what else did I miss? A, B, D, A, big B, small d, A, small b, D. Sorry guys, help me out here. What else am I missing? Thank you. A, V, D, and then I need one more. A, big B, small d. Did we have that one yet? We don't. Okay, so those are the 8 possible outcome, right? 1, 2, 3, 4. Oh no, we just have the same one. So so I'm missing one of the big A one. Sorry, I am not functioning right now. A, B, D, this one. Yeah, that's the missing one. Okay, so total of 8. 1, 2, 3, 4, 5, 6, 7, 8. Okay, but you don't have to list it out every time, right? Like you just have to do the multiplication, right? Okay, to see what the answer is. Is that okay? Do you guys understand how I got 8? Should we do another one just in case? All right, let's say we have AA, BB, CC, DD, EE, and then FF. Okay. How many different possible outcomes for this one? Okay. You can just type the answer in the chat, or you can just, you know, say it out. Okay. Safi and Christian say 8. Anyone else? Do you guys agree, different, or you have a different answer? Okay, so basically anything that's the same, we can just ignore. Okay, and then there are 3 different ones. So we just go 2 times 2 times 2 again, which gives me 8. Okay, make sure you know how to do that. Moving on. Next one, we have the law of segregation. Law of segregation. What does that tell us? Okay, the law of segregation says that only one of the 2 alleles in a diploid organism, which is us, right, will be passed on to each gamete. All right. So just like I mentioned before, the 2 chromosome 1, right? Only one of each chromosome 1 will get passed on to the sperm or egg, which makes sense, right? If you pass on 2 chromosome 1, then when that sperm fertilizes another egg, the zygote will have 3 chromosome 1, right? That's too much. All right, this one is troublesome. Okay, it's a lot of explaining to do. Let me copy it and put it on a new page. Before I do that, does everybody understand the difference between autosomal dominant and autosomal recessive? Do I need to explain that? Just gotta take this stuff away. Autosomal dominant versus autosomal recessive. Does everybody understand that? Okay, so right here. So there is a quick way you can do this without thinking too much. And that is this. Okay, I'm going to put a star here. Autosomal recessive. Okay. A definitive sign that tells you something is autosomal recessive is that 2 parents without the trait, whatever trait you're talking about, okay, has a child with the trait. Okay, that is like a telltale sign that the inheritance pattern is autosomal recessive. If you can find someone with the trait whose parents do not have that trait, then you can say with high confidence it is autosomal recessive. Okay. For autosomal dominant, okay, the if a child has the trait, then at least one of the 2 parents must have the trait. Okay, so let's take a look at here. We have a child with the trait. Being shaded in means you have the trait, and the parents don't have it, right? So that's the thing we're looking for to help us decide whether or not it is autosomal recessive. So this is definitely autosomal recessive. So this person is going to be little a. The parents don't have that trait, so they must have the capital A and then small a. Okay. What about the brothers? Well, they don't have the trait, so they must have at least 1 copy of the big A, but we are not sure about the other one. Okay, the other one could be a small a, could be a big A. There's no way to tell. Okay, so that's the best we could do. Okay, next one. Let's see what it is. Now, this one is a little bit hard to tell. Okay, we don't have a telltale sign like this. Okay, but we can try to, use logic to reason our way through. So let's say we assume it is autosomal recessive. Okay, autosomal recessive. That means everybody who's shaded in, it's going to be little a, little a. Okay, so this is small a, small a, small a, small a. Okay. But that wouldn't make sense, because if they're both small a, then all of the individuals here should also be shaded in, right? They should all be small a, small a. Because if the parents, if the father and the mother only has the small a allele to pass on, then all of their children must also be small a, and we do not see that happening. So for that reason, it cannot be autosomal recessive. Okay, so not recessive because everyone in generation 2 will have the trait, but they don't. Okay, so that's how I know it is not recessive. So the only other possibility is autosomal dominant. So how do we write this? Well, if it's autosomal dominant and none of these people have it, then they must be small a, small a. Okay, which means the parents must be heterozygous. Okay, if they're both big A, then all their kids would have it as well, right? Okay, but they don't. So that means they must be big A, small a. This person has to have a big A, the one that gives him the trait. The other one, we don't know yet. But if we look at their kids, okay, we can see there is a person with the trait. So this person must be small a, small a. One of the small a came from the mother. The other small a, therefore, must came from the father. So for that reason, we know he is big A, small a. Okay. These ones, all big A, small a. Understand? Now, if all of them, if all of the grandkids, if you would, okay, if all of them are shaded in, then there is no way for us to figure out the other half of the father. Okay, it could be another big A. We wouldn't be able to tell. Okay, we're only able to conclude this because they have one child without that trait. Okay. When I talk about trait, it could be like eye colors, right? It could be like blood type. It could be like whether your earlobes are attached or detached, right? So all these are considered as trait. It could be a disease, right? It could be like, sickle cell anemia or something like that. Okay, what about the last one? Anybody want to tell me what you think the last one is going to be based on everything we've said? I'm going to give you a moment. If you think you know, please tell me. Really? Nobody? What if I put this on the test? What if I show you this picture and ask you? Christian said, would Gen 1 want to be small a, and then big A, small a? Could be, but before that, let's figure out what the pattern is. Is this autosomal dominant or is this autosomal recessive? Okay, so, right. Yeah, Safi says recessive. Very good. You have to use this thing that I'm trying to tell you, right? Two parents without the trait has a child with the trait. That's how you know it's recessive. Look, these two people don't have the trait. How do I know they don't have the trait? They're not shaded in, right, on the pedigree. So they have a kid who has it. So that's definitely recessive. Recessive. If this is dominant and this one is recessive as well, right? Okay. So once you figure that out, then this is small a, small a, small a, small a, small a, small a. And then the parents is going to be big A, small a, like that. right? Because that's the only way to have a child that has the trait, right? Same thing over here. Big A, small a, big A, small a. Okay, let's figure out the rest. Okay. This guy, number 6, okay, he is big A for sure. But what about the other half? Do we know definitively what the other half is? Do we know whether it's big A or small a? Anybody? Yeah, we don't know. Okay, we don't know. We don't know. Right? We know they got the big A from one of the parents, and then we don't know what they get for what he got for the other allele. Okay, the only way we'll find out is if he gets married, have some kids, and then maybe we will be able to figure it out. Yeah, exactly right. Because we don't know because he doesn't have any kids yet, right? Okay. All right, over here, same as this guy. No idea, no clue. We'll just give him a big A. Okay. Over here, this daughter, this woman, big A something. Okay, she has a bunch of kids, so we should be able to figure it out. Okay, her husband is small a, small a. Okay, that means all these kids do not have the trait because of the mother. Okay, so all these kids are going to be like big A, small a, big A, small a, big A, small a. Right, so all these small a's came from the father. And all the big A's came from the mother. We don't know. We don't know what the other half is. Okay, so the only way for us to find out is if they had another kid and that kid has the trait. Okay, if they have another kid, okay, and then it's straight in, then we would know that she is heterozygous. Does that make sense? Okay, great. Let's do some of these questions. Any requests for the monohybrid here? If not, I'm just gonna choose one from each section and then do it for you. Okay. I will do number 6, I guess. Number 6 right here. Okay, in humans, brown hair is dominant over blonde hair. If a woman with brown hair whose father has blonde hair marries a man with blonde hair. So let's take a look at this situation. The woman with brown hair, right? Since brown hair is dominant over blonde, so we know the woman is gonna have one big B. But is she big B, or big B, small b? Both of these are brown hair, right? And then small b is the blonde hair. Okay, so how do I know if the woman is big B or big B, small b? Well, fortunately, we know something about her father. Her father has blonde hair. So that means the father, like the woman's father, is small b. So he can only pass on the small b to his daughter. Okay, so this small b, this is from her father. Guarantee? Okay, I don't even know about her mother, but for sure, this small b came from the father because that's the only thing the father can pass on, right? Okay. If you think about it, the father has the small b chromosome. So 100% of all his sperm will be carrying the small b gene. Okay. Guarantee to be passed on. So who is she marrying? She is marrying a man with blonde hair. Okay, so another blonde hair person. This is small b. So if you take the brown hair woman, cross it with the blonde hair guy, what are we gonna get as kids? We're gonna have big B, small b from the woman. So half her egg is going to carry the big B, half her egg is going to carry the small b. And then for the sperm, right, 100% will be small b. And if you do the Punnett square, right, you just match the letters together, and you will see that there is a 50% chance that their child is going to have brown hair. Brunette, right? I guess that's what you call it. And then the other 50% chance of having blonde hair. That's how you do it. We good? All right. Let's do another one. Dihybrid cross. All right. I am going to do number one, and then I'm going to do number two. Dihybrid cross means we look at two traits at the same time. So over here, we have cattles. Either they have long horns or short horns. Yeah, I'll post this. Yeah, it's going to be like in the same place that I post all the recordings, right? Like that Word document. So yeah, you will see this afterwards for sure. So the long horns are dominant over the short horns, and the black coat is dominant over the white coat. Determine the percentage of phenotypes and genotypes of the cross here. Okay, phenotype means what they look like. Genotype means what their genetic makeups are. Okay, so let's take a look here. Okay, we got the small l, big B, small b. We're gonna cross the big L, small b, small b. So can someone tell me what this thing is? Like this, what are we talking about? Cows? Okay, what this cow is that what kind of phenotype does it have? Does it have long horn, short horn, black coat, white coat? What's going on with this cow? Anybody? Okay, thank you, Christian. This is short horn, and which is a black coat. Yeah, so I'm just gonna say short and black. What about this guy? What is this lady cow? I guess. Okay, if this was the I don't know the names of different animals. Apparently, you call them different things if they're male or female. I can never tell. So this is the father cow. What about the mother cow here? Right? That would be the long horn and the white coat. Very good. Okay, so we're gonna see what kind of gametes they're gonna produce. Okay, so it's kind of like math, you know, 2 times x plus 2. You're gonna take the 2 and you distribute it into the bracket, right? I think you learned that in math already. So that's exactly what we're gonna do. We're gonna take each of the letters in the first part here, and we're gonna distribute it to the second part. Okay, so we're gonna get small l, big B, and then we're gonna get small l, small b. And then we're gonna do the same for the other L, but it turns out to be the same thing, right? I'm gonna get another small l, big B. I already have it. I don't need to write it again. Okay, small l, small b. I already have it. I don't need to write it again. Over here, big L, small b. And if I do it again, it's the same thing, so I don't have to write it. And then I will have small l, small b. Does anybody have any question about how I got those gametes? You have to know how to write a gametes. Otherwise, you can't solve the problem. Wonderful. So now we do the Punnett square. Do the Punnett square here. Okay, so half of the sperm is gonna have the small l, big B. The other half is gonna be small l, small b. Half the egg is gonna be the big L, small b, and small l, small b. So when they come together, right, big L, big B, small b. Big L, small b, so on and so forth. Okay. So I will have 25%. 1 out of 4 is 25%. Right? 25% chance of having a big L, small l, big B, small b, which translates into longhorn and black coat. 25% chance of getting this one, which translates to longhorn and white coat. 25% chance of getting this one, which is short horn and black coat. And finally, we would have 25% chance of getting a short horned white coat cow. That's how you do it. Questions? Alrighty, I'm going to do number 2 with you. The answers are all here, right? So like you should do it on your own when you have time. So here we go. We are dealing with plant beans right now, beans. And there are 2 traits we are looking at. Broad pods. Oh, you know what? This is not the question I want to do. This is not the question I want to do. You can do that later. I want to do this one, the rabbit one. If you're with me, you want to write this down in a nice piece of paper. Okay, this one on like a separate piece of paper that you will be able to, have quick access to it during the quiz or something. Okay, and I'll tell you why in a bit. It will save you a lot of time. Okay, so let's read this thing first. In rabbits, we have black fur dominant over brown fur. So big B is black fur, small b, brown fur. The other trait we're looking at is normal length is dominant over short length. Okay, so big N is normal, small n is short. Okay, or called Rex. So what we're going to do is we will diagram a cross between a homozygous black with homozygous normal fur. Okay, and then we're going to cross it with a brown wax rabbit. What does that even mean? Okay, so for homozygous black, so that's big B, big B. Homozygous means same, right? Heterozygous means different. Homozygous black with homozygous normal. And then this other one is going to be small b, small b, small n, small n. So what about the gametes? Okay. What's the gametes from this one? Well, there is really only one possibility. That's big B and big N, right? Over here, there's only one possibility. Small b, small n. You don't really have to do a Punnett square because it's only like one thing, but you could. So you would end up having 100% black normal fur. Okay, so this is what we call the F1 generation, the first generation. Are we okay before we talk more? Sure you don't have any question? Can you scroll up for a second? Sure, I can scroll up for a second. There we go. Okay, now we're gonna take two rabbits from the F1 generation and we are gonna cross them. That's what it says, right? Find the, this is a typo, sorry. Find the ratio, not ration. Okay, ration is like food. Find the ratio for F2 generations. Okay, so F2. How do we get F2? So we're gonna take two of the bunnies from F1 generation and we're gonna cross them. Okay, so let's write out the possible gametes. What are the possible gametes? Over here. The possible gametes are big B, big N, big B, small b, big N, and small b, small n, right? Like this, right? And the exact same thing for this one. So we're gonna have a really big chart. Okay, so I would like you to take a moment to fill this in. I am just gonna run to the bathroom quickly. And then when I'm back, I'll take this up with you. Okay, so please fill this in. All right, so hopefully you get these, right? I'm just gonna combine the letters, big B, big N, big N. And then so on and so forth. You can compare your answers with mine. If you are not sure how I got a particular one, please let me know and I'll explain. Okay, I'm just combining the letters, right? That's the chart. Anybody have any questions? Okay, I hope you get the same thing. Now, if you have some highlighters, you can highlight with me. Okay, let's try to find the ones that are the same. So we looking for anything that has a, let's take a look at the first one here. Big B, big N, big N. So that is like a normal length black coat. Okay, normal length because big N is here, and then black coat because big B is here. So I'm going to look for everything else that has a big B and a big N in it. Okay, so this one, normal length black coat, normal length black coat. These are all normal length black coat. Same thing for these ones. Okay. Are there any other normal length black coat? Did I miss anything? Does anybody see other normal length black coat? Are you guys with me? Are you still there? Okay, my question is, all the ones we highlighted are black coat, normal length fur, right? Because there's a big B and big N in all of them. I'm just asking, are there additional black coat, normal length cow? Yes, where is it? Can you type out the genotype for me, please? Yes, there it is. And there's another one. Very good. Okay, so I think that's all. So in total, we have 9 out of 16. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16 will be normal black coat. Normal black, what do they call it in the question? Fur, yeah, we talk about rabbits, sorry. Normal black fur. Okay. All right, let's find another one. Okay, let's see this guy. What is this thing? This little bunny is black coat, fur, sorry, black fur, but short hair, right? Short black fur. Do we have other short black fur? Right, so take a look. Go find other short black fur. That's it, right here, right? And then right here. Right? And I think that's it. We would have 3 out of 16 short. 3 out of 16 normal. No, short. I'm so sorry. Short because it's small n. And then what color is this? This is brown fur. Black fur. My bad. Sorry. Very sorry. Okay. Yeah. Big B, right? Okay. And then long story short, we have these brown normal length. Okay. 3 out of 16. Normal brown. And then finally, 1 out of 16, which is this guy. 1 out of 16. What is this? Short brown fur. Okay, so we get our 9 to 3 to 3 to 1 ratio. Is that okay? Any concerns here? Whew, that was a lot of work. Okay, I'm sure a question. These are like actual questions I took from the bank, right? So make sure you know how to do it. So here's a question. I'll just read it to you. 2 true breeding stocks. True breeding means homozygous at all genes. Okay. I'll write that down for you. True breeding. Homozygous. On all genes. For all genes. Okay, so one parent has red axial flowers. You don't really have to know what it means. So it's just a trait, right? Okay, axial as in like the flower comes out like here, as opposed to at the end. The other is white and terminal. Okay, so all F1 individuals have red, have red and axial flower. So this tells me that red is dominant over white and axial is dominant over terminal. Okay, because thank you, because of the trait that's being shown up in the first generation. Okay, so we can say big R is red, small r is white, big A is axial, and then small a is terminal. Okay, so they're saying if there are 1,000 F2 offspring, okay, how many of them will be red and terminal? Okay, how many of them will be red and terminal? So if you get this question on the quiz, right? Okay, you are not gonna waste 5 minutes to draw this chart. Okay, we're not going to waste 5 minutes to draw this chart, right? Okay, so you should, that's what I mean, right? Like you should have a good copy of this chart somewhere, I don't know, on your wall, next to your computer, somewhere that you can take a look at it quickly. Okay, so I'm going to put the chart here and see. My goodness, it's not really working. And there it is. I'm just going to pull it right here and we'll see. Okay. So this is the 9:3 to 3:1 ratio. Okay. So if you are looking for F2 generations with the dominant trait, remember, normal and black is the dominant, right? Then it's going to be 9 out of 16. If you're looking for offsprings where it's a mix of a dominant trait with a recessive trait, right? Doesn't matter which way, okay? You can have the dominant for one and recessive for the other, or the other way around, and it's going to be 3 out of 16. If you're looking for 2 recessive traits at the same time, and it's going to be 1 out of 16. So over here, the question asks, there are 1,000 offsprings. Approximately how many of them would you expect to have red and terminal flower? So red is a dominant trait and terminal is a recessive trait, right? Okay, so therefore, which ratio am I looking for? Okay, remember, 9 out of 16 is dominant. 3 out of 16 is 1 dominant plus 1 recessive, and then 1 out of 16 is recessive. Because we're looking for 1 dominant trait combined with a recessive trait, that is going to be 3 out of 16. I don't even have to do the chart, right? I can have the chart in front of me to remind me of which ratio I'm looking for. Yeah, but I don't really need to do the chart myself, right? I don't have to write out the, big R, big A, big R, small a, so on and so forth, right? We can just use this as a reference. So 3 out of 16 times 1,000 offspring. What is that? You're going to need your calculator for the test, okay? So 3,000 divided by 16, which is 187.5. So that's the answer, okay? It's not exactly 187.5, but it's the closest number, right? You might wonder, why is it not 187.5, right? Well, remember, these are just probabilities, right? Okay, when you do 3 over 16, it's like approximately 18.75%, right? Okay, so it's just approximate probability. You're not gonna, just like if I flip a coin 100 times, okay, you are not gonna get exactly 50 heads and 50 tails, right? Okay, you're gonna get somewhere close to that range, but not exactly. So that's why the answer is 187. It's E, 190. Does that make sense? Okay. What if the question asks you for white terminal flowers? How, if you have 1,000 offsprings, how many of them will be white terminal? What would the answer be then for this one? White terminal. I'm going to launch the poll and see if you can do it. Okay. White terminal. The options are the same options here. Okay. Same options. Same question, same options. Okay, so only 2 people answers, and both of the people who have answered got it right. So white is a recessive trait and terminal is a recessive trait. So if it's recessive, then it's 1 out of 16. Okay, so we just have to do 1 out of 16 times 1,000. right? So that is going to be like about 62.5. So this is the answer if we are talking about Y terminal. All right. I hope that makes sense. If all else fails, draw the chart, but it's not the best way to use your time on the test, right? Because, you might have 2 or 3 of these questions. You cannot afford to draw this like 3-4 times, right? Okay, what about this one? In a cross of this, heterozygous ABC, what is the probability of producing the genotype that's shown here? All right. So big A, small a, cross big A, small a. What's the probability of getting big A, big A? Well, that's going to be 25%, okay, or 1/4. You can do the Punnett square to convince yourself. Okay. So there is a 25% chance, a quarter, 1/4 chance of getting a big A, big A. And that's the same for B and C, right? Because they're all heterozygous. So that means the answer is 1/4 times 1/4 times 1/4, which is 1/64. Okay. Let's do one more. Here's the pedigree. Just gonna zoom in. The question asks, what is the likelihood that the children of 4-3, so 4-3 means you look at generation number 4, and then individual number 3 and individual number 4. What's the chance that their kids will have the trait? Okay, the trait is the big W here. Okay, big W. It's a dominant trait if you read the question. So, I mean, we could find out their genotype and see. Right? So individual number 4 does not have the trait. So she must be small w, small w. Individual number 3 must be big W and something. Is he big W, or big W, small w? Heterozygous or homozygous? Well, we will have to trace back to see about his parents. So his parents, the mother is not shaded in. That means she does not have the trait, the woolly hair, okay, whatever that means. So this one is small w, small w. So because of that, he is guaranteed to be heterozygous. Okay. This small w came from the mother. So now if you take the big W, small w, and you cross it with the small w, you will see that there is a 50% chance their child will have the woolly hair. Okay. Okay, let's do more questions. Let's do incomplete dominance. Incomplete dominance. In a type of bird, the heterozygous condition for the allele for black feather and white feather is blue feather. So everything we've done so far is dominant covering the recessive. Okay, but in incomplete dominance, the heterozygous condition is a blend of the two extremes. So let's say you have a red and white flower. The heterozygous condition is going to be pink. Okay, that's called incomplete dominance. Okay, and then there's codominance. So codominance, instead of having a pink flower in the middle, you're going to have a flowers with white petals and red dots on it, for example, right? So rather than having a blend, both of them are being expressed. Okay, so over here it says the bird, we have the black feather, that's big B, big B. We have the white feather. The white feather is small b, small b. And then you have the big B, small b, the heterozygous condition. That would be the blue feather. So what's going to happen if we take a blue bird and then we breed it with a black bird? So blue bird is this. Black bird is big B, big B. When you do the Punnett square, you will see that 50% chance of big B, which is black, and then 50% chance of big B, small b, which is white. Blue. Okay, you do it the same way. It's just that the way you interpret the answers, it's going to be different. Any question? Do I need to do one more or you get it? Okay. One more? Sure. Let's do bluebird, bluebird. Bluebird is big B, small b. Okay, because the question said so, right? The heterozygous condition is blue, right? So big B, small b, and big B, small b. So big B with big B, it's going to be like this. Big B, small b. Big B, small b. Small b, small b. So 25% chance, big B, which is black. 50% chance, big B, small b, which is blue. And then 25% chance, small b, which is white. Is that okay? Okay, good. Let's do a sex-linked question. So a trait is sex-linked if it's carried on the sex chromosome. So either on the X or on the Y. Okay. So let's take a look at this fruit fly question. So in fruit flies, YI is transmitted through this allele, W on the X, and then red eye is the normal one. Okay, so let's write out all the possible combinations. We can have a XX. So this would be a red eye female. I'm just gonna put big F. Red eye female. XX is female, right? XY is male. So you could have a X and then an XW, the allele that gives you a white eye. But because the normal one, the normal red eye is dominant to the white eye, the normal X will cover up the white eye allele here. So this is still a red eye female, but we call her a carrier. A carrier is someone who carries the a certain trait but does not express it. Okay, so even though this fly has the white eye allele, but it doesn't have the white eye. Okay, the red allele covers it up. So the only way for the female fly to have a white eye is XWXW, which is white eye. Okay. What is a normal, what would a red eye male look like, the genetic makeup? Anybody? How should I write it for the red eye male? Yeah, red eye male is XY. Red eye male. And white eye male will be like this. Okay. There's no carrier for male. They only have one X. So, you know, if they have the white eye X, they are going to be white eyed. Okay, so let's try the first one. A here. We have a white eye female and a red eye male. So white eye female is this one. So XWXW. And we're going to cross it with a red eye male, which is like this. Okay, red eye male, white eye female. And we are going to do the Punnett square. Half the sperm will be X, half the sperm will be Y from the male fly. And then for the female fly, all of it would be XW. So when we do the Punnett square, we'll get this result. So 50%, half of the time, you will get a red eye female. And 50% of the chance, you will get a white eye male. Okay. I will do another one. Both Frank and Kate have normal vision. Color blindness is X-linked. Okay, you should know that. I talked about it in the lecture. Their son is tested positive for color blindness. What is the probability that their daughter is colorblind? Okay, so we have Frank here and Kate. Okay, they both have normal vision. So that means Frank is going to be XY, right? And then they have a son and a daughter. The son is colorblind. So we're going to shade him in, colorblind. Okay. So if he's colorblind, what is his genetic makeup? Well, it's going to be X colorblind, Y. Where did he get this colorblind gene? Well, not from the father, we know, because the father has normal vision. The normal vision guarantees the father has a normal X, right? So that means this XC came from the mother. But the mother doesn't have color blindness, right? She has normal vision. So that means she is a carrier. We can color half of the shape for carrier. Okay. So what about the daughter then? Okay. For the daughter, she will inherit 1X from the father. And then the other one is going to be from the mother. Okay, but we don't know. X or XC from mother, we have no idea. Okay, so she might be a carrier. She might not be a carrier, but doesn't matter. The question is asking, what is the probability that their daughter is colorblind? Whether she's a carrier or not, she will not be colorblind. So the probability of their daughter being colorblind is 0%. No need to test her. Does that make sense? Any question? Okay. I will do fill in the blanks here. The genetic makeup of an individual is called genotype. This question asks, what is required to get color blindness? Okay, so for female, color blindness is this. Colorblind. For male is this. Okay, because female, they have two X's, right? So they need to have two of the colorblind allele in order to get color blindness. Okay, because one copy of the X is guaranteed to be normal from the father, it doesn't matter what the other half is. She is guaranteed to have normal vision. Does that answer your question? Okay. The genetic makeup of an individual. No. C doesn't stand for carry. C that's just stand for like color blindness. Okay. If we are talking about like the why I fly, then I could use like W, right? Okay. It's whatever subscript you want, really. So the genetic makeup of an individual is called genotype. The outward appearance with respect to the specific characteristic is called a phenotype. Okay, so you can think of the phenotype as the manifestation, the expression of your genotype. The dominant allele is one that is always expressed if present. An individual is homozygous for a trait if both alleles are identical. If they're different, then it's called heterozygous. Sometimes it's called like pure or hybrid. The recessive allele is only expressed if both copies are present. We talk about incomplete dominance. It's a type of inheritance pattern where neither allele dominates the other and both have an influence on the individual, results in a partial expression of both. It's like the bird, the black and the white feather somehow ends up being blue. If they are both fully expressed, then we call that codominance. Okay, an example is blood type. One person have type A blood, another have type B blood. They're kids, they might have type AB blood. We'll learn that next semester. A trait is a version of a characteristic that is inherited. A pedigree is a diagram, the ones that we've drawn, right? All those squares and circles. That shows the genetic relationship between individuals. That's a pedigree. A gene is a segment of DNA sequence. And the location of this segment is called locus. Right? So the big A, big B, small b, all those things, right? Where do you find them? That's called the locus. A trait that is inherited by the X chromosome is called an X-linked trait. It could be a Y-linked trait as well, but only males would get it. Like baldness, for example. A person that has inherited a recessive allele for a genetic trait or mutation, but usually does not display the trait or show symptoms of the disease, is called carrier. In humans, there are 23 pairs of homologous chromosomes. 23s are autosomal, 1 to 22 are autosomal, okay? And then X and Y, the 23rd pair, that's the sex chromosome. A tetrad is formed when two pairs of homologous chromosomes come together, genetic material between homologous chromosomes are exchanged during crossing over. That happens in prophase number 1. Meiosis produces haploid gametes, sex cells. If the chromosomes don't separate when they're supposed to, if they don't separate during anaphase 1 or anaphase 2, we will get what we call non-disjunction. And that means, you know, some gametes will have extra chromosome, others will be missing chromosomes. That's it for this week. There will be no tutorial next week. Next week, you will work on the lab. The lab is the microscope lab. I'll post a link to it later. The lab is due next Friday, 10 p.m. Make sure you get it in ahead of time. And then we will resume the tutorial the week after. Okay, lecture 9 is probably like the hardest unit lesson of the entire course. Okay, it's a lot of tricky concept. So make sure you watch the recordings early and work on the study guide before you come to the tutorial. Okay, because you know I do go through them quite quickly because I assume you have worked on it already, right? So make sure you do that for the next lecture. We're almost done. We're at week 10. So just 2 more lessons and then it'll be the final test and then you'll be done the semester. Okay, thank you for coming. I'll see you all next week. Have a good weekend. No, I will see you all in 2 weeks. No tutorial next week. Sorry. Bye.